Hello everyone, and good afternoon, good morning, good evening, wherever you are, and welcome to today's Twist-hosted webinar and roundtable discussion on addressing the challenges of high-throughput SARS-CoV testing. My name is Chris Thorne, and I'm the Senior Manager for Field Marketing here at Twist. So before we begin, just a little bit of housekeeping. As you've noticed, your microphones aren't switched on, so we'll keep all lines muted during the webinar. We will be having questions, and actually we'll be having what I hope is an interesting discussion after the presentations. So if you do have questions, please use the Q&A box at the bottom of your screen rather than submitting things in the chat. And finally, once the event culminates today, once you leave, we'll be sending you over to a brief survey, and we'd be very grateful for any feedback that you can give us by filling that in. Okay, so the format for today's webinar, if you like, is slightly different from our previous webinars in that I'm very excited to have two speakers joining us today presenting on the topic of addressing the challenge of high-throughput SARS-CoV testing. And I'd really like to encourage everyone to stay until the end, as once we've done the presentations, our speakers will then be joined by Rebecca Nugent, Twist Director of R&D, for what should be a really insightful roundtable style Q&A afterwards. So as I said, if you have questions, please do submit them as we go along in the Q&A box. So the first of our speakers today will be Jo van der Swampler, who is the CSO and founder of Biogazelle, a CRO specializing in high-value genomics applications to support pharmaceutical research, clinical trials, and diagnostic test development. Jo is also a professor in functional cancer genomics and applied bioinformatics at Ghent University, Belgium, and is a world-renowned expert in the domain of RNA quantification and non-coding RNA. Our second speaker today will be James Grayson. James is from UgenTec. James is lead field application specialist for the Americas at UgenTec, where he brings over 10 years of experience automating laboratory workflows to that role. So prior to joining UgenTec, James was the head of molecular workflow automation at Siemens Healthineers, where he worked to capture customer workflow needs and develop customized solutions. Okay, so Jo, I'm going to hand over to you now and let you take it from here.
Thanks very much, Chris. I hope you can hear me okay.
Yeah, we can hear you just fine.
All right. So thanks very much for the kind introduction. I'm happy to be here. Let's just start off. Awaiting a vaccine to prevent or medicine to cure, the key priority is to prevent its spreading of this virus. So we can do this by physical distancing, washing our hands, wearing a face mask when appropriate. But this has to be combined with massive PCR testing to detect and to isolate infected individuals. So Biogazelle holds a unique forefront position, as already said, in the application of quantitative PCR, digital PCR, and RNA sequencing. And we are an ISO 17025 accredited contract research organization for qPCR test development and use in clinical trials. So the company was founded on a revolutionary method for qPCR normalization called GNorm and data analysis, QBase+, with more than 20,000 citations and thousands of customers worldwide. We have co-authored the MICI guidelines for design, execution, analysis, and reporting of qPCR studies, again with more than 10,000 citations thus far. And we have wet lab validated more than 100,000 qPCR assays beyond industry standards, and the SARS-CoV-2 test is just one of them. And since April, we're also part of the Corona Standards Working Group, an initiative from JIMB, the Joint Initiative for Meteorology and Biology from Stanford University, dealing with the development and provision of access to standards, control materials, inter-lab comparisons, and knowledge to perform accurate SARS-CoV-2 tests. And the ultimate goal there is to develop and to build a COVID-19 diagnostic standards development partnership. So the SARS-CoV-2 RT-qPCR test workflow consists of 4 main analytical steps, here indicated in pink and executed by Biogazelle. It involves the transfer of the primary sample tube to a 96 well plate to increase downstream throughput, followed by a viral RNA purification, RT-qPCR detection of the virus, and data analysis and authorization. Important to note is that the entire workflow is pretty complex and is in fact a multi-party workflow involving healthcare professionals, logistic partners, or clinical labs to make the test kits, the swab and the tube available, to distribute it, to collect it and bring it back to the lab. but also IT partners for sample registration, reporting of results to the doctor, etc. And if possible, do all this within 24 hours. To visualize the complex workflow, a nice 3-minute video was made, which I'm not going to display here, but the link will be shared by the moderator for you to watch after this webinar. So in the midst of the worldwide supply chain issues of reagents and instruments, we set up a scalable platform in end of March, beginning of April, in less than 2 weeks, adhering to a set of unique design principles. And this was only possible because of our expertise in PCR test development and use in clinical trials. First of all, we wanted our platform to be flexible and modular, such that different components from different suppliers could work together, each optimized for their specific part of the workflow. And modules can be interchanged, and we use automation where it can make a difference. Of course, the platform had to be high throughput with the aim of at least 6,000 samples a day and smart scalable. This means that we introduce more units of a given module or more instruments within a module if needed. In terms of business continuity, we have established a strategic stock, standing orders, drop shipments, and validated alternative suppliers. We really needed a dedicated procurement officer to manage all this. For instance, for the RT-qPCR mix and the RNA extraction, we have 2 validated suppliers to mitigate any potential supply chain issues. And I think that's critically important to keep your system 24/7 up and running. We also wanted our platform to be autonomous, so no A to Z commercial solution or a black box, of which most of them are relatively slow or generally face still worldwide shortages for instruments and consumables. And finally, our platform had to be high quality by default, but built for constant innovation and improvements through internal cross-validation. And I will explain this later during my presentation. Our platform effectively consists of 16 instruments to process 6,000 samples a day. And for further scaling, we don't really need to linearly increase each instrument. For instance, our qPCR setup robot, number 1 in pink, we have one, it can handle more samples than we actually need. For qPCR, we have not used the stacking modules of our qPCR instruments, but we have validated if need be. Extraction scales per 2,000 samples per centrifuge, and this is exactly what we call modular, flexible, and smart scaling. So the most challenging step in the entire workflow is actually the transfer from the primary sample tube to the 96 well plate for downstream high throughput processing in a standard SBS format, so 8 rows times 12 columns. Not only because this is a very time-consuming process, but also because of safety concerns. We're working with the live and potentially harmful virus. The inside and the outside of the tube may be contaminated with this virus, posing a biosafety risk for the operator. Therefore, everything is processed within or by skilled operators in so-called biosafety cabinet class 2 cabinets. One team of 3 operators can operate in a shift of 4 hours for processing 470 samples in 3 cabinets. Processing means the transfer of an aliquot of the swab transport medium to a deep well plate, followed by the addition of lysis buffer, which is the first step in the RNA extraction. And from that moment onwards, the virus is inactivated and does no longer pose any health risk for the operator. To process 3,000 samples in an 8-hour working day, we need 18 operators and 9 safety cabinets. Because of the highly focused work, shifts are limited to 4 hours. So to reduce the dependency of qualified manual operators, we have also introduced automation at this step. Our TCAN robotic system has a barcode scanner to scan the patient tubes and the destination plate, obviously, and an 8-channel liquid handling module with liquid sensing to aspirate from the patient tube and dispense in the 96 well plate. The plate has been pre-filled with a lysis buffer using 96 multi-channel arm on the system. This robot can single-handedly, well, not really because we need 3 operators, but anyway, do it by itself, 3,000 tubes in an 8-hour shift by only 3 operators. And this is a reduction by more than 6-fold compared to the manual workflow. Importantly, fine-tuning is critically required to handle the swaps inside the tubes and the very viscous nature of these samples. So it's not just plug and play, it requires some tuning to have this level of automation. When we come to the next step, RNA extraction, to select a suitable kit, we evaluated cost, ease of use, throughput, guaranteed availability, and of course, performance. We really went to great length at evaluating various kits and doing so on a standardized set of viral stock dilutions and patient samples in 4 different transport media, which are representative for what's available outside, including liquid amines buffer, phosphate buffered saline solution, etc. The results from our analysis were quite shocking, with different RNA extraction methods showing greatly different performance. The transport buffers also differed greatly, with clear interactions between the transport buffer and the RNA extraction kit. On the left, I show you serial dilutions of this inactivated viral stock from 10 to the minus 3 to 10 to the minus 5 for only 2 RNA extraction kits, but 4 different transport media. CQ values in the y-axis and the log 10 of the dilution in the x-axis. What you can see here is that the linearity and the slopes are excellent, but the intercept values, where the curves cross the y-axis, are varying greatly. This means a great difference in detection sensitivity by more than 30-fold or 5 PCR cycles. So for RNA extraction, we finally settled on filter plates from either Norgem Biotech or Zymo Research, processed in a simple centrifuge. We use a centrifuge with 4 positions for deep well plates, and the Epimotion 96XL dispenser is used to transfer the lysis to the filter plates, and the ViaFill is used for quick plate dispensing and washing with elution buffers. For washing, and adding the elution buffer. The Mantis is used to dispense 4 microliters of spike-in control and carrier RNA. And this carrier RNA is really essential to improve extraction efficiency, especially for the lowly concentrated samples. One operator that is qualified and trained in our lab can process 374 samples in about 1.5 hours. 3 operators can thus process 6,000 samples in an 8-hour working day. Of note, other automated or semi-automated solutions do exist, but they either require much higher investments or do not reach the same throughput. So for qPCR, we use a 384-well pipetting robot or 96-well pipetting robot to dispense in 384-well qPCR plates. We use again a Tecan system where we dispense 40 microliters of master mix with 6 microliters of sample in a 20 microliter total volume. Normally we do qPCR in much smaller volumes, but to maximize detection sensitivity, we aim for maximum volume. We have 2 validated master mix solutions, as already indicated. It's Biorad's iTech mix and the Takara Bio PrimeScript 3 as validated and highly sensitive one-step RT-qPCR mixes. The actual qPCR is done in 384 well plate, as already mentioned, on a CFX 384 with stacking modules, but we don't need the stacking modules with the current throughput of 6,000 samples a day. Data analysis obviously is a huge challenge, and the next speaker will for sure elaborate on that. You can imagine that if you have to look at 6,000 individual curves on a single day and interpret them for diagnostic accuracy, you need support. We use FastFinder from UgenTec that allows automated data transfer from the instrument to the UgenTec cloud, uniform interpretation with a little bit of artificial intelligence, and importantly, numerous quality control checks. It looks for positive and negative controls. It looks at the value of the internal spike-in control, does trend analysis, neighborhood analysis to detect potential cross-contaminated samples. And the data analysis occurs at 2 levels. There is the interpretation by the data reviewer, and then the authorization level to finally send out the data to the doctor or the clinical responsible. Importantly, the software is coupled to our internal LIMS system or to the LIMS system of the laboratory or the hospital. Our platform is approved by the Federal Institute for Public Health, Cienciano, and the Federal Agency for Medicines and Health Products, FEMHP. And we also operate in an ISO 17025 lab environment where we have filed accreditation or asked accreditation for our test, which is waiting an audit end of the month and approval, hopefully. We also participated in European quality assessment schemes and processed proficiency samples from our customers. To safeguard our quality, we have multiple controls in each experiment, namely an internal spike-in control to control for RNA extraction and RT-qPCR of each individual sample, and 2 positive and 2 negative controls in each batch of 96 samples processed in a 96 well plate. Finally, we have also introduced digital PCR as an orthogonal validation method and to calibrate our platform. Talking about control samples, in the next 3 slides, I want to illustrate how we use the Twist Biosciences control RNA in our workflow. First, we use it as a positive control for RT-qPCR, as already mentioned, whereby we spike about 1,000 molecules in well H12 bottom right in each plate. It not only serves as a positive control, but can also be used for trend analysis and interrun or interplate calibration if needed. Secondly, during assay development, one of the experiments, we created a serial dilution series of this positive control RNA from 25,000 copies down to 1 copy in a constant background of our spike-in control, followed obviously by duplex RT-qPCR testing. This does not only allow to determine linearity and coefficient as the coefficient of determination of such a serial dilution series, but also RT-qPCR efficiency based on the slope of the curve and the single molecule CQ value, which is 35, about 35 in our experiments. It also allows to determine the limit of detection, which is the lowest concentration at which 95% of the technical replicates are detected. So on the right, you see a visual representation of 32 replicates of 6, 3, and 1 cDNA copies and 0 copies as a negative control. And as expected, we did not detect any signal in the negative controls. These are the bottom or the right 2 columns. And for 6 copies, we have perfect detection, 100% of the 32 replicates. However, if we look in a closer view at 3 copies, we see that we lose signal in 5 wells. So our limit of detection is between 3 and 6 copies, where we have at least 95% of the samples detected using this particular qPCR mix and our eGene assay, obviously. So this is really tailored towards each individual's platform. And finally, we use the Twist positive control RNA also for qualification of our qPCR instruments to demonstrate fit for purpose and equivalence of the diagnostic test over the different instruments. Amongst others, with testing a serial dilution series, but also a homogeneity plate in which we test 384 identical samples as shown on the right side. We want to see good linearity, efficiency, reproducible detection of the limit of detection, but also uniform CQ calling and endpoint fluorescence values. Of note, we have a free app that is available on our website that can process these types of data from a qPCR instrument and actually does a functional validation of any qPCR instrument. Also, the link for that will be shared by the moderator. And to finish my part, I want to show what ongoing developments we have in place. Despite its relatively slow mutation rate, SARS-CoV-2 accumulates mutations while it persists in the human population. And it is important to evaluate PCR-based diagnostic assays as new SARS-CoV-2 genome sequences become available. So based on the COVID-19 genome analytics in Edge Bioinformatics from Los Alamos National Laboratory in the USA, 99.5% of more than 50,000 SARS-CoV-2 genomes known today will be detected by the eGene assay. A little bit smaller, 98%, will be detected by the NGene assay. So here you see the added value of combining the E and the NGene, because it not only slightly increases detection sensitivity, but it also significantly increases strain detection coverage up to 99.99%. And finally, the Belgian Institute for Public Health, CNSANO, estimates that early next year, January, February, Belgium will face 40,000 daily consults of flu-like symptoms for a period of about 100 days, based on the data from prior years. For a population of 11 million inhabitants, this is an equivalent of 1 in 275 individuals. Most of these patients with flu-like symptoms will hopefully be free of SARS-CoV-2, but when doing a test to rule out SARS-CoV-2, it would of course prove valuable to determine if the patient is infected with influenza or respiratory syncytial virus. We are therefore developing a 7-target, 4-color multiplex assay to detect SARS-CoV-2, influenza A and B, and RSV A and B. And with that, I want to conclude. We have set up a high-throughput modular SARS-CoV-2 RT-qPCR detection platform. Primary patient sample handling is the major bottleneck, followed by RNA purification. The analytical measurements, meaning the qPCR, scale really best. And we have built in a customized, or we have built a customized solution that is scalable, has low cost, and ensures business continuity by all the features that we have built into our platform. Finally, control samples are key for assay development and quality assurance. I want to end by acknowledging and thanking some people and institutes, the Belgian task force, obviously, for PCR testing, several companies that have contributed instruments, such as Formulatrix, BASF, the Innovation Center in Ghent, Biorad, Ghent University, Imbiose, CIMIT, and Janssen, and finally, the entire COVID-19 team at Biogazelle. With that, I give the word to the next speaker and look forward to the discussion.
All right. Thank you, Yo, and hi, everyone. Let me just share my screen here. So, hi, and thanks again, Yo, for taking that. For the next few minutes, we're going to take a look at some of the complexity of high-throughput testing environments and at some of the tools that can be used to manage that complexity. Well, over the last few months, we have all seen and experienced various levels of response to the COVID-19 pandemic. And over the last few months, UgenTec has been fortunate enough to have been involved in supporting both local and national initiatives for COVID testing. What is interesting in this sort of emergency situation that we're in are the challenges of rapid scale up to enable in some cases, countrywide level testing. Some of these complexities that we've experienced can include the variability that comes from different initiatives having to use various instrument providers and manufacturers, so having a mixed population of testing devices, variable assay kits from different providers, so having both variable kits themselves, but those kits might have some variability in the genes that they're targeting, and have variability in their own control systems as well. And we definitely see a challenge in the availability of trained clinical staff to interpret and report results. And implementing standardized methods of measuring and verifying lab performance across multiple locations is critical in these cases. In the following slides, we're going to show how you can manage some of this complexity using instrument and assay agnostic intelligent software solutions. Sorry, oh, there we go, sorry. So to address some of the challenges of high throughput COVID screening, UgenTec's FastFinder platform has been used to support large-scale testing initiatives. And this FastFinder solution is comprised of analysis. And analysis is an instrument-agnostic, assay-agnostic PCR data analysis and results reporting solution. Analysis does curve evaluation, results reporting, and decision tree application, which is the automation of your assay interpretation rules directly from your IFU or SOP. This is critical in these cases because in this high throughput workflow where you may be supporting more than one assay, more than one kit, you may have various interpretation rules, and we can digitize that to make sure that everything is being applied correctly. And with that, we take clinical reporting far beyond standard instrument software. Now my keys are working. Sorry. Next, we have workflow. Workflow is your laboratory's air traffic control system. So this allows you to manage instruments and track samples through your testing process. And we'll talk more about that in just a bit. Lastly, we have Insights. And FastFinder Insights is your laboratory's operational performance tracking system. Insights provides standardized dashboarding and detailed lab intelligence that visualizes key performance parameters like positivity rates and for critical targets, QC performance across instrument fleets and sites, and testing turnaround time. All of this solution, as Yo mentioned, is a cloud-based software solution, and we are securely hosted and highly scalable because of Microsoft Azure and our partnership there. We are ISO 13484 compliant and ISO 27001 compliant. We're going to dig a little bit deeper into FastFinder analysis. So analysis is the primary component that is being used and was highlighted by Yo. Analysis is our artificial intelligence-driven data assistance, data management assistance. So in a typical workflow, in a typical lab condition, we see users having to interact with the instrument software, having to review, manually review and interpret PCR amplification curves, cross-referencing those against various flags that may or may not exist in the software, and cross-checking those against the rules and interpretation from either the IFU or the SOP. The idea behind FastFinder is we can digitize all of that and really just make that a one-click or fully automated action to go from raw data into reportable answer out. Part of how this works for us is we apply artificial intelligence to interpret the PCR curve. So in the simple, straightforward case, we have sample thresholding, which most instruments on the market use to determine a CT or CQ. In the perfectly textbook case, that's very applicable. But what I think we've all experienced who have worked in the industry is that PCR curves can be far from textbook. And so we use artificial intelligence to look at not a threshold, but the various features of the curve, the slopes, just the behaviors overall, how things might have artifacts from instruments or chemistries or workflows, and take that into account in interpreting and producing, interpreting the curve and producing either a CQ and finally a call for that curve. Part of our process is training of the data set, which can be very specific to a given instrument or assay. And it's easy to see that you could potentially call a negative and a clean positive a positive, but what do you do with a gray zone, and how do you manage curves that aren't behaving in a textbook fashion? And that's part of what our software does, in that we can train the artificial intelligence algorithms to interpret curves that are clearly negative, but could be something like a creeping curve with an upward drift. And do you treat that as a negative, and what you do with that? Here we see examples of a software interface, and the important thing to see here is that, and to understand, is the software is agnostic. So in this COVID case, in these emergency cases, we can support a significant number of instruments and from many different instrument manufacturers, all with one interface, and still have that same feel and touch that some people are used to, as far as being able to see my curves, see a plate layout. And then what we do on top of that, using the application of the decision tree, is to be able to then combine the results, do the analysis, and compare that against all of the validity criteria for the interpretation. And in doing that, we can generate a final result and also flag any results that are atypical or anomalous, or something that the end user would like to have called out for them and flagged for manual review. And this is how we can support both the lack of availability of trained staff and standardize behavior of interpretation in having variable human factors involved in making a final call. On the right here, we see some idea of how we build in this interpretation, our decision tree builder, how we can add rules and various CQ ranges to performance, and that's something we can help and use to capture the rules of the IFU. Next, we're going to speak a little bit about workflow, and workflow is your sample chain of custody across plates, instruments, and assays. So with workflow, we're taking an entire look at all of the instruments involved in taking a sample from raw primary tube in to final result out. And we can capture various workflows and present them in a dashboard. So we can capture all of the steps of the workflow, all of the instruments involved. If possible, we can speak to those instruments and dashboard performance across the entire workflow and through the entire testing process. So we can have views of recent analysis, logs of any issues or errors, and just really have an idea from a dashboarding perspective of how the lab's operating and what's currently, what steps any given sample can currently be in. We can take that down to a very detailed level within workflow where we can track an individual sample through its whole process in the lab, and we can do this in an automated way. So we can see when a sample is first transferred from the primary tube to the plate. We can track that through extraction and all the way through PCR analysis result. And we have chain of custody and audit trailing through the whole process. We can do that both in an automated fashion or here in a manual fashion. So some instrumentation in these workflows are not automated, maybe do not produce files, and we can add different audit tracking and trailing through the testing process to at least confirm that steps have occurred. Another key feature of FastFinder is insights. And insights is the operational business intelligence of the laboratory. So we can use insights on top of FastFinder workflow and analysis to generate automated reports and verify the performance of instruments and operators and sites. And we can do things which I think are critical here, like monitoring batch quality with control systems. With insights, we can provide standardized lab intelligence, in which we can provide, we can monitor things like testing rates, positivity rates and other key performance indicators for the lab. In this case, we could track over time the positivity rate of COVID that's passing through the lab. We can see the number of tests and performance, number of tests, number of instruments, number of sites involved in a lab. And we can monitor the individual behavior of controls, like the positive control or negative control. Negative control, not so much, I guess. This is an example of the dashboard that can be used to track performance across the lab. Here we see that we have full access to a multiple site testing, and everything is sort of grayed out for privacy purposes. But we can see the performance of individual labs across a multi-site testing initiative. We can see what instruments these labs are using and what rate. We can see different assays that are being tested, and all this can be monitored remotely from a central location if there's an overseeing body. Here we see, and something that I think is very critical to the overall concept of a national initiative such as COVID testing, where we can see the behavior of positive controls over time by instrument, by target, and really look for variation. We can flag deviation in behavior from the system. And this is something that can be used for troubleshooting, for monitoring individual performance at sites. It's something that has been used in various proficiency monitoring programs that are implemented in some of these testing initiatives to do ongoing QC of the lab and across sites and initiatives. Sorry. In conclusion, Compiling and implementing in a high throughput testing program comes with significant challenges in resource availability and resource management and program standardization and management. And we have worked with many high volume initiatives to mitigate those challenges with intelligent software. And thank you for your time, and back to you, Chris.
Thank you, James, and thank you, Yo. That was two very interesting talks, so very much appreciated. Looking forward to discussing a little further now. But before we get into that, I just want to begin by welcoming Rebecca Nugent, who joins us today from California and is Twist Bioscience's Research and Development Director. She joined Twist after spending many years in biofuels and green chemicals industry and leads our R&D teams focused on the development of synthetic biology and next generation sequencing target enrichment products. So Rebecca, welcome and thank you for joining us.
Hi, thanks for having me.
Okay, great. So I'm just, so I just want to begin, if I may, with a general question. So we've seen two quite technical presentations there, and I think it highlights the amount of work that's gone into getting this all set up in the last few months. But I guess we weren't here, 9 months ago. None of us were working on COVID, and I think we've had to move at speed to make this happen. And I wondered, and Yo, perhaps I could direct this question to you to begin with. There's a tension, I guess, here with dealing with this pandemic between needing to get these workflows set up at speed and making sure that we can, address this need as quickly as possible, with needing to be as rigorous as possible in terms of the workflows that we're using. And I wonder if you could just, maybe just comment on whether you have seen that tension in terms of the work that you've been doing, and how you guys have sort of approached dealing with that.
Yeah, excellent question. Indeed, we experienced a lot of tension and pressure. So setting it up in less than 2 weeks, obviously we cannot get to the same diagnostic quality as a lab that has been doing viral diagnostics for 20 years. If they implement a novel test for this new virus, that's much easier to have it embedded in a lab with experience to do so. So what can you do is controls, and validate every step as good as possible. And I think we have to be honest that we improved over time. It's impossible to do that in 2 weeks' time. But having control samples and knowing what to expect when something is negative, when your sensitivity is at stake or specificity, etc., it allows you to see how well you're doing. Exchange samples. That's what we did very early on with the labs that were already up and running. Do some kind of ring testing, exchange samples, participate in proficiency schemes, and learn and see where you can improve. So it's an ongoing activity, and I think we're still doing that. We can always perfect, we can always improve on many different aspects, and we have done so the last 4 or 5 months. So it's an ongoing activity to learn and to improve your diagnostic test.
Yeah. And Rebecca, perhaps I might point that question to you as well. One of the sort of underlying principles of SynBio, right, is this process of design, build, test, learn. Can that still be applied in the context of a pandemic? Do we have that luxury, or do we have to get it right first time, do you think?
That's a really interesting question. Yes, I think you can. I think Yo just answered that question, that you set your baseline, you do your first experiment, and then you see how you can iterate and improve on that first experiment. Yo's response really resonated with something that we did at Twist, which was originally when we developed the synthetic controls, we went from concept to commercial launch in about 3 weeks. We've never done that before. It was an incredible effort. And so what we learned from our first iteration of building our 2 control strains, we then applied that knowledge to the other controls that we released, which are just genetic variants of SARS-CoV-2. And then we took that knowledge and we applied it to more controls for different viruses with the respiratory controls. So yes, there is room for the design, build, learn, test cycle, even at the pace in which we're moving at this pandemic.
Fantastic. And James, one of the slides which sort of jumped out at me was towards the end of your presentation, where you showed how your software allows you to look across multiple labs potentially, but using the same software. So has it been your observation that that these labs are able to how are labs learning from one another? Are you able to feed back when things aren't going quite so well? Is there a sort of collective effort to improve that you're seeing from your perspective as a
there is, and that comes in a couple of different ways, but definitely what we're seeing that works is these proficiency monitoring programs. So we see an overriding sort of monitoring QC program that is put in place using various controls to really track how individual labs are performing, and then using that data to see how to make improvements to find maybe issues with systems. We've been asked to do things like flag for different error types and bring those to people's attention, and so I think it's a really interesting mechanism for optimizing performance, for truly trying to get to the best quality of result using the variable both instruments and reagents that are available in this situation.
And if I may add to that, it's amazing what you can learn by looking at lots of data. Trends that you simply don't notice, first of all, if you don't have the tools, but with massive amounts of data, you see things that you didn't see before.
Well, for example, do you have an example in mind? I'm just curious.
Sure. Repeated freeze-thaw cycles of reagents, a new primer that was purchased synthesized. You see performance difference if you accrue that over time, and you have many measurements with that same lot, something that remained unnoticed to us before.
And in fact, that leads me on to another question, which is you show the use of the controls in your 96 well plates as a sort of in-process control. But do you see there being a need but you obviously have a multi-stage workflow. Are there controls applied across the workflow, or are there gaps that you think there would be a need for different controls to get that level of oversight?
It's a good point. We can discuss a lot about controls, the ideal controls. I think there are hundreds of different control materials available, and there are also different schools of thought here. We definitely have workflow controls from the very beginning. Our negative controls, they're really the sample transfer is a negative sample that really goes to the primary sample transfer, RNA extraction, RT-qPCR, data interpretation. But then we include an additional negative control, and our positive controls in the very beginning, we only used the positive control from you guys, which came after RNA extraction, which is an RT-qPCR control and a data analysis control, but it didn't control for extraction. So that's why we have patient samples now, highly positive patient samples that we dilute and that we introduce in each plate as a more representative sample that undergo primary transfer, extraction, and RT-qPCR. So I think there is room for controls at each step of the workflow. And it's important to consider that not all controls control everything you want, and that's why you need to build in multiple controls.
Understood. So
you mentioned negative. Oh, sorry, Chris. Go ahead.
I'm sorry, Rebecca. No, please. Rebecca.
You mentioned negative controls. Can you go more into the importance or the differentiation between a positive control and a negative control, and the type of considerations that go into defining what those actually are?
Is this a tricky question?
No, it shouldn't be a tricky question. I mean, so from my perspective, go ahead, but I can clarify more if you'd like.
Well, our negative controls, I'm not sure if they are the best ones or if you could consider other options, but for us, it's a water control in the same buffer as the patient sample, because the matrix has an effect as well. The ideal negative control would, of course, be a patient sample, which is confirmed to be negative, because then everything, also the human RNA in a sample, may have a specific matrix effect. It's more challenging and difficult to do so, so we use water as a negative control. But I think the most important here is to show that you don't see signal when you don't expect one. So false positives are clearly a huge problem, and they come from two sources. It's reagents, and it's well known, it's documented. Many of the suppliers have worked hard on that their reagents, primers, probes, PCR mix, extraction reagents even, were contaminated by probably ramping up their production by synthesizing so many synthetic template to cater for the entire world, that all the, I think, production facilities, not all, but many of them were contaminated with SARS-CoV-2 template. So you need controls to make sure that your lab, your reagents are not contaminated, but also cross-contamination is a genuine concern, especially in a high-throughput lab. I know it should not be there, we don't want it, but some of these patients have such a high viral load, billions of molecules actually in there. So the tiny drop of a femtoliter can create a positive signal in a neighboring well. That's why you need controls to try to capture them. We do that at 2 levels, our negative controls in the process, but also using the software that will flag a warning if 2 positive samples are adjacent to each other, which alerts us, and we can then make a call whether we should send them for repeat extraction or not.
I mean, that's exactly what I was looking for. When we were developing our respiratory controls, one of the things that we were really keen to demonstrate was that it could be used as a positive control to isolate or detect viruses like influenza and Ebola virus. But we also made sure that it was a negative control for SARS-CoV-2 because cross-contamination is an issue. Sensitive detection is also a great issue to raise. I love your point right there of talking about cross-contamination and how you use the software. I guess, James, a question for you, or just to shift this over to the software side, what kind of flags or warnings do you look at when you were talking about the gray zones for those samples that may be positive or may be negative? Do you go into that data when you find 2 positive results next to each other?
Sure. It's a little bit of a 2-part question based off of how you asked it. First is, interpreting the positive itself. So is this positive? Is it, is it clearly a positive from a normal sample? Is this a late positive that could be related to a false positive result? So something that really amplifies really late. Then we do a proximity evaluation. So we will look at the space around a high positive and a low positive and see if there's any chance that this could have been a local cross-contamination, and we will raise that flag as one of many different flags that we can put in place to bring attention to the reviewer to check this. And that's really built into something that we call the decision tree, which is working with the end user to build a set of logic. How do you want this reported? If we see this morphology, if we see this pattern, how do you want us to treat it? Do you want us to bring it to your attention? Do you want to immediately invalidate that result and send that back to retest? So it is a lot of working in collaboration with the end user to make sure that we've captured how they would like to interpret data within the software itself. But using the intelligence, we can capture all the information in flags and do like a smart cross-check. Does it make sense that this could be a cross-contamination from a local neighbor? And so that's, I don't know. To me, that's the really cool part about the analysis and the intelligence. So
I guess off the back of that series of questions about what we've sort of how we've come to this place, I want to ask all 3 of you whether you feel that some of the learnings that you've taken from the last 6 months are going to be things that you can take forward, both in the context of your current work, but also into different disease areas as well. So perhaps if I could begin with Yo. So obviously, Biogazelle's been doing this kind of work for a number of years, but perhaps not at this scale. Do you see yourself continuing in this manner, or what are the things that you as a company want to take forward from hopefully when the pandemic is behind us?
But to make it clear, we are qPCR experts. We design, develop, validate qPCR, digital PCR tests. However, we have never done diagnostics before, and that's a really different ballgame. Not only the massive scale that we are operating under, but also the more strict requirements of turnaround time and quality, or guaranteed quality, as doing a molecular diagnostic test is really different. So we definitely have learned a new way of doing this in high throughput, short turnaround times, with superior quality that a molecular diagnostics lab or clinical lab really demands. And this is the experience that we want to bring to our clinical trials. We do a lot of clinical trials for our pharmaceutical or biotech partners, but this is also very similar. Perhaps the turnaround times are not that strict in a clinical trial, but often the throughput and the quality is very on par. And we definitely have learned a lot of lessons that we can bring over to the clinical trial division. Also, our LIMS system was, I would say, not perfect before, and we had to make it perfect today to track every single detail from a new dilution. And this is also very helpful for future work.
Okay. And James, what about you? So is the FastFinder software just applicable to infectious disease, or will you be expanding, or perhaps it's already applicable?
Yeah, so FastFinder, in the before times, before COVID, FastFinder was very, it is a clinical diagnostic tool. So we started FastFinder, and the analysis portion of it really focused on the clinical market and results interpretation and reporting. But before all of this, we had also grown into different areas like oncology, PGX. We support ag bio, veterinary testing, and a lot of different applications and a lot of different modalities, not just RT-PCR, but melt curve and various other PCR-like adjacent analyses. So I think, from that side, it's nice to be out in all of these different industries. From specifically this, the COVID pandemic, I think it's been, as a software solution, as a clinical solution, it's been good to see that we could scale up enough to reach, hundreds of thousands of analysis per day globally with some of the programs that we're involved in. And what I really see us developing over these last months, though, has been the insight software. So it's been that business intelligence layer to really track performance across multiple sites, across multiple locations and labs and modalities that I think has been critical to and hopefully we can continue to implement for whatever comes next. And I think that's where I'm very proud of UgenTec and my small part in it. And, small part to play in the whole pandemic as well. So it's been good to be involved.
And Rebecca, so, Twist is a DNA synthesis company, and here we are making RNA. So tell me, what are we taking forward as far as you're concerned, and your team's learnings as well?
Yeah, we learned how cross-functional our platform is, and how just how ubiquitous DNA is as a tool. DNA is a starting material for a variety of different tools. And in this case, we used DNA as the template to make the synthetic viral controls. We also used our platform to create next generation target enrichment products so that we could detect and characterize different viral genomes. So I think the takeaway that we've learned from here is our ability to use our platform in a very speedy way to respond to the challenges and the threats that our society faces. And then also has a tool we can use, target enrichment, and then these synthetic controls for detection and characterization of emerging pathogens or viral threats.
Brilliant. Right, so I've tried to sort of thread in a few questions from our audience as we've gone along, but there's just a couple of technical ones which I think I'd like to address. So perhaps one for James. A question from one of our participants: how much data is being used to train FastFinder? And I guess this is a good question to ask now, because perhaps you're getting more data than you would have been 6 months ago. So is this also proven an opportunity to improve the artificial intelligence algorithms, or perhaps you could speak a little bit to that?
Yeah, and to be clear, this is not my personal area of expertise, the artificial intelligence and all of that. But we've been on the market with FastFinder with the artificial intelligence and curve interpretation for 3 to 4 years, if not more. I kind of lose track sometimes. So we have really good out-of-box performance, right? So it's for interpreting those curves, it's, we're at a very high level of accuracy where we train and where we try to pick up on performance and where there's always room to grow is various workflows and various chemistries and various instruments that are all strung together. So some of those things can, I think Yo had mentioned just the behavior of curves with different kits paired with different buffers and how that variation can lead to different morphologies and curves. And that's where we really target and work with the individual workflow and the individual users of that workflow to capture the expert's interpretation of those corner cases. And that's where we're always learning and always picking up is how do you pull in those unique corner cases that aren't the perfect PCR curve.
And I have a question here for Rebecca regarding the controls from Ali Bektas. So just wondering if you could go into a little bit more detail about how the synthetic controls are themselves QC'd. So obviously we want to supply them to Yo so that he can use them out the tube. So what are we doing to ensure that our tubes are as we say they are?
There are multiple QC steps along our process. One of the QC steps that we use is next generation sequencing to confirm that the sequence of the viral genome is what we think it is. We also use qPCR, and we're transitioning into digital PCR to confirm the relative or the approximate amount of molecules that we're distributing into each tube that we deliver to the customer. So one, sequence verification, and then two, verification of the number of copies per microliter that we're shipping out.
Brilliant. Right, well, we're coming up on the hour, so I want to kind of wrap things up just by saying, first of all, a big thank you to our speakers today, James and Yo, and also thank you, Rebecca, for joining us today. On a personal level, it really has been astonishing to me to see how the scientific community has risen to this challenge, and it's really fun to actually dig into a specific case and see how these three organizations have sort of collaborated, if you like, to actually make a big difference. And I can imagine that that's happening in any number of labs the world over. So I really appreciate being able to take your guys' time today and just dig into this particular use case. I will just say to our audience, thank you for joining us today and just sitting with us as we talk through these challenges and see how they've been addressed. If you want to follow up with us, we'll be posting the recording of the discussion and the presentations on our website in due course. Please do take a minute or so once we leave the webinar just to fill in our survey and give us some feedback. That'd be very much appreciated. If you liked this format, let us know and we can do more of the same. And again, a big thank you to Rebecca, James, and Yo, and hopefully we'll see you all at the next one. So thank you, guys.
Thank you, Chris.
Welcome.
Thank you very much.